أعزائي طلبة المرحلة الأولى في كلية الطب جامعة كربلاء، إليكم المحاضرة الأخرى من محاضرات الفصل الدراسي الثاني العام الدراسي 2020-2021 للفصل الدراسي الثاني كما ذكرت، وتتعلق المحاضرة بالهكسوز مونوفوسفات شنت. Definition of hexose monophosphate shunt, or the other name is called pentose phosphate pathway. What are the biochemical rules of hexose monophosphate shunt? Hexose monophosphate shunt constitute of two different general stages. What are the sources of NADPH, coenzyme or nicotine amide, adenine dinucleotide phosphate, reduced form coenzyme, and what are the biochemical rule of this coenzyme? What is the rate limiting step or rate controlling step of hexose monophosphate shunt, and how the hexose monophosphate shunt is regulated? And the other very important point of hexose monophosphate shunt is the genetic defect of glucose 6-phosphate dehydrogenase or G6PD deficiency, which is the rate limiting enzyme of hexose monophosphate shunt. Hexose monophosphate shunt is very important. Started with glucose 6-phosphate. The HMP shunt is an alternative pathway to glycolysis in which about 30% of glucose molecule enter the HMP shunt and the remaining 70% enter the glycolytic pathway. The biochemical rule of hexose monophosphate shunt. Very important rules are performed by hexose monophosphate shunt. First, generation of the coenzyme NADPH and various pentoses, 5-carbon sugars, as well as ribose 5-phosphate, which is very important for the biosynthesis of a nucleotide, that's mean purine and pyrimidine bases. Purine and pyrimidine bases, as you know, is very important for biosynthesis of nucleic acid. The hexose monophosphate shunt is very important also in red blood cells or in erythrocytes. What are the stages of hexose monophosphate shunt? We have two different stages. The first one is called irreversible oxidative phase or reaction. The reducing equivalent NADPH and ribose 5-phosphate are generated in this phase or in this stage. It provides a mechanism for the conversion of pentose sugar to triose and hexose intermediate of glycolysis. No ATP molecule, no adenosine triphosphate is directly consumed or utilized by this pathway. The oxidative portion of the pentose phosphate pathway, as indicated in the following figure, consists of 3 irreversible reactions that lead to the formation of ribulose 5-phosphate, which is the final product of this phase after 3 enzymatic reactions, and 2 molecules of reducing equivalent NADPH are produced. The HMP-SHUNT is very important for the mammary gland and liver, lactating mammary gland, and adipose tissue for NADPH generation, because various enzymes in these tissues are dependent upon the presence of the reducing equivalent NADPH molecule. For example, in the testes, ovaries, placenta, and adrenal cortex, NADPH dependent reactions are required for biosynthesis of steroid hormones, and in red blood cells for the NADPH dependent reduction of antioxidant glutathione. Glutathione is very important endogenous antioxidant. The body system must be maintained. يجب أن يكون في حالة توازن ما بين الأوكسيدنت and antioxidant. That's mean generation of oxidant biomolecules, including free radicals such as superoxide free radicals or hydroxyl free radicals, and non-free radical molecules such as hydrogen peroxide. These are oxidant molecules. These are produced normally and they must be removed. The removal of these oxidant is by the presence of various antioxidant molecules, including exogenous antioxidant that must be provided daily, or by dietary materials such as vitamin C, vitamin E, vitamin A, and so on, selenium metal ions, okay? And we have another types of endogenous antioxidant such as enzymatic and non-enzymatic. The most important non-enzymatic antioxidant is the glutathione, which must be present as reduced glutathione GSH. This figure indicates the overall reactions of HMP-SHUNT as shown in this figure. We have 2 different stages. The first stage, which include oxidative reactions, which must be irreversible As shown, glucose 6-phosphate is converted into 6-phosphogluconate and liberation of first molecule of NADPH as indicated فوق السهم. هذا التفاعل يشتمل على two enzymatic reactions, and the second irreversible or the third irreversible اللي هو أيضا عملية تحول 6-phosphogluconate into ribulose 5-phosphate and generation of a second molecule of NADPH. تلاحظون بالخطوة الأولى generation of NADPH, the first molecule تشتمل على two reactions, and the second generation of NADPH اللي هي بالخطوة الثالثة اللي هي أيضا تكون irreversible. هذا ما يتعلق ب the irreversible oxidative reactions. الانزيمز 1 and 2 as indicated جوا السهم المقصود بها 6-phosphate dehydrogenase and 6-phosphogluconolactone hydrolase. Two reactions, two enzymatic reactions, and the third enzyme اللي هو catalyzes the generation of ribulose 5-phosphate and a second molecule of NADPH هو 6-phosphogluconate dehydrogenase. The other reactions include the non-oxidative reversible reactions, that mean interconversions between ribulose 5-phosphate as indicated بالتفاعل reversible number 4, number 5, number 6, number 7 إلى آخره. تلاحظون various carbon number derivatives compounds, saccharide compound are produced, including ribose 5-phosphate, pseudoheptulose 7-phosphate, erythrose 4-phosphate. تلاحظون 5 carbon atoms, 7 carbon intermediate, 4 carbons, xylulose 5-phosphate, glyceraldehyde 3-phosphate, 3 carbon intermediate, fructose 6-phosphate, okay, fructose 6-phosphate. أيضا we have another molecule of glyceraldehyde 3-phosphate and xylulose 5-phosphate by transketolase and transaldolase or transketolase enzymes that are dependent upon thiamine pyrophosphate coenzyme derived from vitamin B1 thiamine. The glucose 6-phosphate dehydrogenase is the rate limiting enzyme that catalyzes the oxidation of glucose 6-phosphate into 6-phosphogluconolactone as the coenzyme NADPH get reduction to reduced NADPH into يعني NADPH plus the oxidized يتحول إلى NADPH with hydrogen, that's mean reduced coenzyme form. And then G6PD catalyzes the rate limiting step, that's mean the first reaction or the slowest or the rate controlling step of HMP-SHUNT. Okay? The initial reaction is the committed, rate limiting, and required regulate the first step of the pathway. Therefore, when the amount of NADPH, the reduced coenzyme, which is the direct product of this reaction, is elevated in the cell, for example, this compound acts as a competitive inhibitor of the enzyme G6PD. إذا صعد تركيزه أو الليفل مالته يعمل كمثبط تنافسي للإنزيم G6PD الذي يعتبر rate limiting enzyme. Okay? Where the amount of NADPH to NADP plus this ratio is sufficiently high to substantially inhibit the enzyme under most metabolic conditions. هذه النسبة decide the rate limiting step of the HMP shunt. The increased demand for NADPH, that's mean when the NADPH to NADP positive, when this ratio is decreased and flux through the pathway increased in response to the enhanced activity of the enzyme. عندما تقل هذه النسبة يتحفز الإنزيم لإنتاج another NADPH product حتى يزداد تركيزه لأنه الخلايا تحتاج as reductive compound and as very important antioxidant biomolecule. Okay, زين. Insulin hormone. Insulin upregulate expression of the gene which is responsible for the G6PD biosynthesis, and flux through the pathway increase in the absorptive state. This reaction produced firstly one molecule of NADPH with hydrogen ion, and then another molecule is produced as indicated in the previous figure. %30 of the oxidation of glucose in the liver occur via the pentose phosphate pathway. RBCs utilize the reaction for production of NADPH by HMP shunt because large amount of HMP shunt is required by the RBCs to maintain the amount of reduced glutathione as endogenous antioxidant. The second phase is the reversible or called non-oxidative phase or reactions, as shown in this figure. تلاحظون various byproduct as mentioned in the previous figure واضحة عندكم. تلاحظون 6 phosphogluconate يتكون من عنده irreversibly ribulose 5 phosphate and from ribulose 5 phosphate various sugar ribose 5 phosphate xylulose 5 phosphate pseudoheptalose 7 phosphate and so on. Various sugar molecules was produced by the second reversible phase of Exos monophosphate shunt, and as mentioned, transketolase coenzyme dependent thiamine pyrophosphate, transaldolase enzyme dependent upon thiamine pyrophosphate are required for these interconversion reactions of the second phase non-oxidative stage. Regulation of HMP-SHUNT, as mentioned, the regulation was controlled by the NADPH, NADP positive ratio, which regulate the activity of the enzyme G6PD, and insulin also regulate the activity of this enzyme. Importance of NADPH coenzyme. The coenzyme NADPH يختلف عن نظيره NADH. NADH هذا الكوانزيم يدخل في اللي ناتج by Krebs cycle واللي يتكون by various reactions يدخل أو carry 2 electrons as hydrogen ion as hydride ion تنتقل للسيتوكروم chain for ATP production. بينما الكوانزيم NADPH لا يدخل respiratory chain or cytochrome chain for ATP production, وإنما يستعمل utilized as a reductive compound, reductive product, reducing agent نحتاجه in various reductive reactions. Okay, and ADPH is required, for example, in the following reactions. Role of NADPH in WBC and bacterial phagocytosis play an important role in bacterial killing by phagocytosis. Okay, mechanism as indicated in this figure. تلاحظون in this figure we have generation of various reactive oxygen species اللي هي for example as shown oxygen, superoxide oxygen, H2O2, hydrogen peroxide, hypochlorous acid. All of these by NADPH as shown بالفيجر هذا 3 اللي يتكون from HMP-SHUNT pathway بوجود glucose 6 phosphate dehydrogenase المسؤول عن generation of NADPH. تلاحظون NADPH oxidase يحول بالماكروفاج اوكي يحول الاكسجين الى super oxide radical species, and then hydrogen peroxide, and then to hypochlorous acid, which is used for bacterial killing. طلاب تلاحظون هذه ال rule of NADPH أو the reducing equivalent اللي هو the most important antioxidant molecule. شوفون الرول مالته وعلاقة ال HMP shunt و G6PD activity وأهميته في إنتاج ال NADPH وعلاقة ذلك في ال bacterial killing in macrophage. أوكي، هذا نسميه respiratory burst, very important biochemical reaction. يتكون تلاحظون from oxygen بوجود NADPH oxidase، يتكون سوبر اوكسايد، والسوبر اوكسايد ديسميوتيز SOD. اوكي، هذا الفيري امبورتنت enzymatic او نسميه endogenous enzymatic reaction، له دور كبير في ازاله تاثير السوبر اوكسايد ديسميوتيز. بالفيجر الثانيه باللون البينك كلر فيجر 4 انا سويتها لون تو كلرز حتى يبين الشرح للفيجر. فيجر 4 نفس الشيء، هو الرول اوف NADPH in bacterial killing وعلاقة ال HMP shunt. تلاحظون هذا الفيجر الجواني in yellow color وعلاقته مع ال killing bacteria. اوكي، هذه واحدة من أهمية، هذا نسميه respiratory burst. وعلاقة ال respiratory burst مع الفاجوسايتوسيس اوكي بالماكروفاج. تلاحظون يتكون هيدروجين بيروكسيد يتكون خلال هذا التفاعل، وبعد ذلك يتكون هايبوكلورس اسيد اوف ايروبيك ميتابوليزم. هنا بالفيجر هذا يوضح ايضا بعض التفاعلات formation of reactive intermediate from oxygen. الالكترونز تنتقل من الأكسجين ويتكون سوبر أوكسايد، ثم هيدروجين بيروكسيد، ثم هيدروكسيل راديكال، وبعد ذلك يتكون ال H2O. وأهمية هذه production of various oxidants وعلاقته مع ال endogenous enzymatic and non-enzymatic antioxidant mechanism. والرول اوف جلوتاثيون واضح بالفيجر الثاني. جلوتاثيون ريديوزد، تشوفون تأثيره هو السكافنجر، يعتبر سكافنجر او act to remove the hydrogen peroxide. اوكي، ويتحول الى ال H2O، وبنفس الوقت يتحول الريديوزد جلوتاثيون إلى أوكسيدايزد جلوتاثيون. هذا كبداية للمحاضرة، حتى بساعة توصلكم بعد هاي المقطع راح نعطيكم المقطع الثاني لتكملة محاضرة ال HMP shunt. أوكي، شكرا جزيلا.